Hello, I'm Chris McCarthy from the American Chemical Society, and welcome to this news briefing from ACS Spring 2021. We're joined today by Dr. Jennifer Lee and Dr. Dexter Dean of the National Institutes of Health. They're studying an amyloid link between Parkinson's disease and melanoma. Dr. Lee?
Oh, thank you, Chris. I'm excited to be here today to tell you about our research efforts to develop molecular understanding of the disease connections between Parkinson's disease and melanoma. In the last 20 years, it's been known that PD patients have a higher incidence of melanoma compared to healthy individuals. And recent studies also indicate melanoma patients are more at risk to develop Parkinson's disease. So we currently do not know how or why these two different diseases are connected. I became interested by this connection because of the protein alpha-synuclein, which my lab has studied in the past decade in the context of PD, may be involved. Curiously, in melanoma, alpha-synuclein levels are elevated, and its levels are inversely correlated with melanin content, suggesting that there may be a relationship or alpha-synuclein could be modulating or dysregulating melanin biosynthesis. So this is relevant because melanin is a protective pigment in our skin and eyes, which absorb UV light, which is a risk factor for melanoma. So in this work, we're presenting a novel hypothesis involving the amyloid formation of alpha-synuclein and PML17 as a molecular link between PD and melanoma. Amyloid formation of alpha-synuclein is essential in PD pathology, whereas PML17 amyloids are important for pigmentation. So I'm going to turn it to my postdoc, Dexter, who's going to tell you more about our major findings from this work.
Thanks, Jen, and thank you all for coming today. When I started this work, one of the first important questions to address was whether or not alpha-synuclein actually localized in melanoma cells to the site of melanin biosynthesis, which is this organelle called the melanosome. So it had been shown that alpha-synuclein was present in melanoma cells, but it was unknown as to whether it was actually present within this organelle, the melanosome. So using cultured human melanoma cells, I applied two different but complementary methods to show that alpha-synuclein was in fact present in melanosomes alongside with PML, meaning that the two proteins had the potential to interact. So I then explored that interaction using purified alpha-synuclein protein and a purified fragment of PML, which forms amyloid, called the repeat domain, or RPT for short. So what I found was that in an aggregated amyloid state, alpha-synuclein accelerated PML RPT aggregation to form a fibril structure that it normally doesn't adopt on its own. And in a non-aggregated, non-amyloid state, alpha-synuclein inhibited PML RPT aggregation altogether. So to summarize, what we found was that alpha-synuclein localizes to the melanosome alongside PML in human melanoma cells and can modulate PML RPT aggregation in vitro, which suggests that the interplay between alpha-synuclein and PML may be one of the underlying mechanisms as to how alpha-synuclein disrupts melanin biosynthesis in melanoma cells, which, like Jen mentioned, may contribute to melanoma progression. And this is what I'll be presenting later today at the meeting.
Great. Thank you so much. Are there any questions from our viewers? Please state your name and affiliation in the chat window when you ask your questions. And I'll start with a question. Is there a difference in the severity of melanoma that Parkinson's disease patients have versus the general public?
That is a good question, Chris. I'm not. Dexter, do you have specific information on that? I'm not aware of any.
Yeah, I'll say in general, there are not very many studies that have actually looked at melanoma in terms of the Parkinson's population. But there are a few that have shown that among PD patients, synuclein seems to be more aggressive than the tumors are more aggressive. They're depigmented, like I mentioned, so they lack melanin. So it could be because the tumor is not pigmented that it goes undetected for longer, which leads to a more aggressive cancer.
Great. So is there currently a predictor for the skin cancer in Parkinson's patients?
Again, that's a very important question, but this is not our focus and our expertise. So I'm not sure in terms of whether alpha-synuclein could be used as a biomarker for melanoma, if that's what perhaps you were thinking about.
Yeah.
But what is clear, though, alpha-synuclein is a good indicator for Parkinson's, and people are exploring that.
I'll just add that there are studies now where they have detected synuclein in the skin of healthy individuals, individuals with Parkinson's, as well as individuals with melanoma. So there is some suggestions that synuclein deposition within the skin could be a biomarker for Parkinson's and potentially also melanoma.
Thank you. So is it known what function, if any, the alpha-synuclein serves in healthy melanin-producing cells?
That is exactly what we would be interested in exploring next in our future, to define the biological role of alpha-synuclein in hopes to learn more about it. But as far as I am aware, currently, that's sort of the mystery that warrants further investigation.
And you mentioned that you had found, you'd used some techniques to kind of see where the PML and alpha-synuclein kind of lined up together. What were those techniques?
Dexter, go ahead.
Yeah, sure. So we used a technique called confocal immunofluorescence. So you use antibodies that specifically detect either alpha-synuclein or PML, and then that antibody is labeled with a fluorophore that you can visualize under a microscope. So essentially, you visualize the fluorescence from the antibody that detects alpha-synuclein. You visualize the fluorescence, which is a different chromophore, which detects PML. And then using data software, you can see where there are overlap of those two fluorescent signals. The other technique we used was Western blotting. So we fractionated human melanoma cells into different fractions containing either nuclear enriched proteins, cytosolic enriched proteins, or melanosome enriched proteins. And that's where we saw that synuclein was present both in the nuclear, cytosolic, and melanosomal enriched fractions. And that was, again, using antibodies to specifically detect alpha-synuclein.
Thank you. What are the biological differences between alpha-synuclein and its amyloid and non-amyloid forms?
Do you want, I can take care of this one. So I guess, I'm sorry, you were asking about biological function of the soluble versus amyloid, or you?
Just sort of the differences, yeah.
Right. So in its soluble state, the protein is intrinsically disordered, or that it doesn't adopt any native fold. And current thoughts in terms of its function in neurons is that may help in assisting synaptic vesicle transmission. Now, in its amyloid state, traditionally it's associated with Parkinson's disease. That's a classical hallmark. Now, what the difference is, that is a self-assembly of alpha-synuclein monomers into these larger nanoscopic filamentous material. The underlying molecular structure are quite different. They're highly enriched beta sheets, and they form the cross beta structure, which has beta sheets running perpendicular to the fibril axis. So those are really distinctive structural features, and the presence of amyloids and disease that is still widely debated in terms of what specific mechanisms, but they are classical hallmarks, and they can be cytotoxic in tissue culture.
Thank you. So do you think that alpha-synuclein could have other effects on the melanoma cells other than its interactions with PML?
We do, and I think this is sort of the tip of the iceberg that we are just exploring. There are others in the literature. There are other suggestions on what may be doing, although limited. So we definitely think that there are other potential mechanisms that need to be explored. Dexter, do you have anything specific you would like to add?
I would just absolutely agree with that. Yeah, we've identified one particular interaction that may be present in melanoma cells, but I really think that synuclein is doing a lot fundamentally in melanoma cells to promote melanoma proliferation, which is exactly what we're interested in investigating in the future.
If someone suddenly starts producing less melanin in their skin cells, are they more likely to get melanoma?
I think that's the sort of, I would agree with that statement, because the pigment in general, that is thought to be protective against UV damage. So that would be the likely result. But I have to say, I can't speak medically and confirm that, but I think that is true.
Let's see. Is there a way that these results could be applied to help Parkinson's patients reduce their chances of developing skin cancer?
Yeah, I'm very happy you asked that question. And this is avenues that both Dexter and I are deeply interested in thinking about, how these molecular connections, how we build upon that. And but currently, much more work is needed to sort of enable to see whether this could be developing into interventional approaches or novel strategies in changing disease phenotype. So we're definitely interested in thinking about that. And I think one thing that we want to understand, hopefully by understanding how alpha-synuclein behaves in melanoma, that may also help us understand potential, discover new ways that it may help Parkinson's disease. Because most of the work has been studied in neurons in a specific context, but that doesn't really explain what it's doing in melanoma cells. So I think this is sort of give us a very interesting opportunity to be able to see whether through our work on melanoma that can teach us something about Parkinson's disease.
Thank you. So what are the next steps for this research?
Dexter, want to take that one?
Yeah, sure. So we're still investigating the interaction between synuclein and PML-RPT in the soluble state. So I mentioned that we've seen that synuclein inhibits PML-RPT aggregation in its soluble state, and we're still trying to explore that in depth. That's something that we've observed, but we want to know the details of. So we're collaborating to collect NMR to get more amino acid residue specific information. And then beyond that, kind of the next step beyond that is, like we mentioned, to more fully understand the functional role of alpha-synuclein in promoting cell survival in melanoma, which we have some ideas of things that we can pursue. We think that it might be regulating gene expression. So we want to do some experiments along those lines.
Thank you. So what would be a take-home message about this research you would want to leave with our viewers?
Do you want to, Dexter, start with the specific findings? Maybe I'll discuss more broadly what we hope to achieve.
Yeah, to just reemphasize our specific findings, and what I'll be presenting today is that alpha-synuclein is present within the melanosome of melanoma cells, which is the site of melanin biosynthesis. And it inhibits that process by potentially interacting with PML and interfering its amyloid formation, or modulating its amyloid formation disrupting melanin biosynthesis, which could potentially lead to melanoma progression.
Yeah, and for me, sort of for a more broadly view is I think that, these seemingly very disparate diseases, yet there is a correlation that gives sort of an opportunity for us to kind of stop and think about the problem differently. And so I kind of, what I like to convey is that to kind of think about things outside sort of the canonical models and try to explore different opportunities that can help us learn about specific mechanisms in human health. And so this is kind of, I think there are many other examples in biomedical research where diseases are connected, but through fundamental research, we learn about things that are very basic, and it would give us insights into future directions. So I think that's sort of a greater take-home message, because it is sort of curious. Why are we looking at Parkinson's and melanoma? But I think that's sort of the underlying motivation and the driving force behind that, to develop fundamental understanding of the molecules that are involved.
Thank you so much, Dr. Lee and Dr. Dean. The archived version of this session will soon be posted at www.acs.org/ACS Spring 2021 Conferences. And please join us for our next press conference tomorrow at 11 a.m. Eastern. Thank you.